This question is from the chapter Principles of Inheritance and Variation from class 12th textbook. Question 122, NEET 2023. Frequency of recombination between gene pairs on the same chromosome as a measure of the distance between genes to map their position on the chromosome was used for the first time. Now, this is a straightforward question. The questions on scientists and their contribution. So, this is something which you have to by heart and remember. Make a list of it. The answer for this question is Alfred Sturtvant. He was the student of Morgan. This is the answer. Let's have a look on what he did. So, the model organism on which he worked were Drosophila. So, map genes to map their position. So, he designed a technique in which he could actually locate where the genes are with respect to each other. The original work on linkage was done by his mentor Morgan. Morgan is credited with the work of linkage. So, this is the location of the gene coding for yellow body. This is the location of the gene coding for white eye. This is for miniature wing. 3 different characters. Another fly with brown body. This was yellow body, this was brown body. This is white eye, this is red eye. And miniature wing. So, when a cross was done, if you do a dihybrid focusing only on the body color and the eye color, you would expect to get the parental types BR, brown body red eye, yellow body white eye. These are called the parental types. And the non-parental or the recombinants would be yellow body and red eye, YR. Similarly, BW would be recombinant, brown eye and brown body and white eye. These would be recombinants. So what they observed was, what specifically Morgan observed was, the percentage of recombinants were very less and parental combinations were high. And that's how he came up with the concept that if the genes are closely arranged on the same chromosome, the parental types will be seen in the progeny because the genes are linked. Linked means they tend to be inherited together. So based on this, Sturtevant, he came up with the concept of mapping. Why not map them based on the percentage of recombinations they show? So if recombination is high, then you would get high percentage of recombinants, and that would in turn indicate their relative distance between those genes. So gene position, which is called the mapping, was directly proportional to the recombinance. Recombination. So if the distance between the genes is high, the percentage of recombinants will be high. So this is the concept. It was used for the first time by Alfred Sturtvant. I hope I was able to clarify you and give you a clear picture on this concept. Thank you so much for watching this video.